Hi there, my name is Chiara Forrester, and I'm a PhD candidate at the University of Colorado in Boulder. Here I'm at Nylaut Ridge, which is near Nederland, Colorado, which is also home of the SU Boulder Mountain Research Station. And here we have this National Biosphere Reserve, where we have a National Science Foundation funded program site with the Long-Term Ecological Research Program. And we make the snow melt earlier by spreading black sand on the surface of the snow at peak snowpack, and then we spread it again on our control plot. So here is where we have this big experiment going where we're looking at how early snowmelt will affect lots of ecosystem processes, and I mostly focus on plant population dynamics and the timing of plant life stages. So in this plot, it goes to 10 by 40 meters, so you can look up here, that's marked by those two corner poles all the way down, and you can see the silver reservoir down past those trees. We have melted out the snow earlier, and it typically melts out about 4 to 14 days earlier. And then we have these warming chambers that are passive warming chambers, so they just warm the soil beneath them about 3 degrees Celsius, which is great for climate change projections. And then we have these control pots next to them. And so in total at this site, we can implement a few different treatments. We have early snowmelt alone, so that's this control plot, early snowmelt plus warming, so we've melted out the snow earlier and we've warmed it. And then on the other side, in our control plot, we have the control plot only and warming alone. And then I've marked, as you can see here, about 900 plants permanently across the tundra. I look at each individual plant twice a week throughout the growing season, and I mark which life stage it's at. So that could be green up, flowering, or senescence, with multiple different stages within those groupings. And I also count how many flowers that plant has to try to understand how early snowmelt, warming, and the combination of the two will affect the timing of those life stages as well as their reproduction. We also record the height and leaf number of all individual plants each year, so we can understand how their growth and biomass may change with the climate. In each plot, we also have temperature sensors that record hourly temperature throughout the growing season. They look like this. And then we also put in plant nutrient probes that track the plant available nutrients such as nitrate, calcium, phosphorus, potassium, to understand how abiotic factors like nutrient content of the soil will change with those changing climate factors. We're currently in year 2 out of 3 of this research, but so far we've found that early snowmelt caused increases in nitrate and calcium availability, but no change in soil moisture. So far we're not seeing that early snowmelt is causing any serious negative environmental impacts on these plants that we're studying in particular. Green up and flowering stages of phenology responded more strongly to early snowmelt and warming than senescence, or when the plants went dormant at the end of the season. Changes in the timing of snowmelt influenced phenology more strongly than warming alone. So we know that these systems are very strongly dominated and controlled by patterns of snowfall and snowmelt, and this is supported here. Early snowmelt has not yet affected plant reproduction or growth, but we're expecting to see lag effects in next year's data because some of these species tend to preform their buds and leaf tissues multiple years in advance, meaning that we might be seeing growth now that reflects environmental conditions of up to 3 years ago. We've also seen that recruitment of Alpine avens, so the germination of new seedlings that we experimentally added, has so far been negatively impacted by early snowmelt and warming treatments, which leads to some implications for the recruitment and perseverance of these species in the future. With that, I'd like to thank the Indian Peaks Wilderness Alliance for supporting this work, and I'd love to talk further about this research with anyone that's interested. Thank you so much for your time.